Cellendes is a company that sells hydrogels for 3D cell culture. I founded this with Helmut Wurst in 2009, and since 2010 we are selling our products to the worldwide market. We also have several distributors in quite a few countries. Our hydrogels are composed mainly of polymers, or sometimes also of highly purified proteins that are cross-linked to make a meshwork. And they are also modified with adhesion peptides or enzymatic cleavage sites to mimic the native matrix. So we produce these hydrogels because we realized there is a need in the field of cell biology and especially 3D cell culture for chemically defined hydrogels. So researchers are able to control the cell environment and also get reproducible results. In this way can adapt the cell environment to the individual requirements of the cell and the culture conditions.
We were approached by Francesco Pampaloni at the Goethe University in Frankfurt, who initiated the project and was looking for a company with a good competence in hydrogel development. Since the LSFM4LIFE project focuses on the development for us, on the development of a hydrogel for the culture of pancreatic cells, we needed to adjust our hydrogel system to the culture of these cells. And then we, of course, also set up culture conditions for making pancreatic organoids here, which really brought us forwards towards the general goal of providing hydrogels for organoid cultures.
First we started with the mouse organoids, so this was pretty easy. We found very quickly a gel that supported their growth and expansion. When we started with the human organoids, this was quite a different story. So it took us a while to find out which adhesion peptide, for example, would support their survival and their growth.
The GMP process, which is required for producing pharmaceutical compounds, is a very controlled process. You need to control everything, and with our hydrogels that is particularly easy, or not easy I should say, it's doable, because since we're using these chemically or biotechnologically produced hydrogels, we know how they are being produced, under which conditions, and also reproduce that production from time to time or each time. And so that makes it much easier to get all these controls done for GMP compatible production. Cell culture has been for decades done on flat surfaces, and now we all say goodbye flat biology, because almost every researcher now, when he does serious cell culture, will want to cultivate these cells in 3 dimensions, and for that hydrogels is a very, very good tool.
Yeah, I think hydrogels will play a big role in the future in research as well as also in pharmaceutical industries, for example, for drug development, because especially these chemically defined hydrogels are much more reliable in their composition. They are controllable, the results are more reproducible. And also in regenerative medicine, we believe they will be applied more and more in the future.